Just going to pick up where we left off on, what was it, Monday. Last thing that we got to talk about was the movement of food from our oral cavity, so our mouth, into our pharynx, and then into our stomach. A fancy way to describe the act of swallowing or deglutition, which, as we learned, is aided by a process called peristalsis. Today we're going to talk a lot more about peristalsis in other places. One of those places includes our stomach. Now, you'll see the word a lot here, and that word is gastric. The word gastric is always going to refer to stomach. We've got gastric pits and gastric glands, gastric juice. So just get ready for the word gastric a whole bunch. Our stomach is a portion of our digestive tube that will dilate or expand into an elongated pouch-like structure located in the upper part of our abdominal cavity at our liver and diaphragm. It contains gastric glands, which contain ducts, can channel secretions of their cells along the ducts into the lumen of the stomach. Several of those gastric glands open into the bottom of, and I should say of, not or, of narrow channels called gastric glands. Okay. Here on the right, we show the structure of the stomach wall. Remember, the stomach is part of the tract, and we learned that the tract from our esophagus to the anus is made up of 4 layers: a mucosa, submucosa, and a muscularis. And then what we didn't really worry too much about in this class was the serosa. Okay. The gastric pits are these indentations of the mucosa and submucosa. Down here in our picture B, we show what a gastric pit looks like. Okay. Basically, it's a way to increase surface area for more cells. Okay. So we can have more cells that secrete various things. Okay. We have 2 different types of mucus cells, surface mucus and mucous neck cells. They do the same thing, we just give them different names because they're located in 2 different places. They're going to secrete mucus. Okay. Mucus in our stomach is absolutely important because of what is also produced in our stomach, produced by what we call parietal cells, and that is hydrochloric acid. Hydrochloric acid is a very dangerous acid, very low pH. And if we didn't have a very thick layer of mucus in the inside of our stomach, that hydrochloric acid would burn right through the wall of our stomach. If you ever get hydrochloric acid spilling up into your esophagus, we call that acid reflux, you know how painful that can be. If you ever dealt with hydrochloric acid in the chemistry lab, it's very dangerous to get under skin. It can burn you. It can burn your respiratory tract if you breathe any of the vapors in. But our stomach is adapted to protect against this hydrochloric acid. Okay. Now, this hydrochloric acid plays a very important role, and I'll talk about that here in a second. Parietal cells also secrete intrinsic factor, which is a chemical needed for the absorption of vitamin B12. We have chief cells that secrete 2 enzymes, okay, pepsin and gastric lipase. Now, pepsin is not directly released by those chief cells. What is released is pepsinogen, a precursor to pepsin. So our first enzyme under proteins is pepsin. Pepsin is an old school named enzyme. It doesn't end in ase like what we see with the amylase and lipase, but it is an enzyme. Okay. Now, gastric lipase is an enzyme that is an ACE. And just like lingual lipase, we've got gastric lipase. Actually, both gastric lipase and lingual lipase work really well in our stomach because they prefer that really low pH. The other day we listed off salivary amylase, which so far is our bone carbohydrate digester. We mentioned that it inactivates, or it becomes inactivated in our stomach. Okay, whereas lingual lipase became activated in our stomach. Okay. Now, that's not the only reason why hydrochloric acid is important. I'll talk about that here more in a second. Other stuff released within our gastric pits include histamine and serotonin by way of enterochromaffin-like cells, or ECL cells. Okay. Histamine, along with gastrin, a hormone released by G cells, helps to stimulate the release of that hydrochloric acid. I'll show you a figure and demonstrate that here in a second. Okay. And then gastric cells secrete ghrelin. Ghrelin's another hormone. This is what promotes us to be hungry when our stomach is empty. Mainly what our stomach does is digestion. It does allow for some absorption, some short-chain fatty acids, some water, some ions. We can actually absorb some drugs like aspirin and alcohol, okay, which explains why it doesn't take several hours for those to take place or to have an effect in our body. Okay. Table 24.3 is a summary of different things occurring in our stomach. It lists off cells that are part of the mucosa and what they do, what we just talked about. We haven't talked about the muscularis yet, but we're getting ready to on the very next slide. And we're going to talk more about the pyloric sphincter, which if I go back to the picture here, here is our total stomach. The pyloric sphincter is the sphincter that allows for the stomach to empty. Okay. And it does so in a very coordinated manner, and we'll talk about it more here in a few slides. Let's first talk about the role of the muscularis, providing us with that peristalsis again that we talked about the other day, like squeezing a tube of toothpaste from the bottom always towards the cap. So right away after food enters our stomach, within minutes, waves of peristalsis pass over the stomach every 15 to 25 seconds. What we do is start those contractions in the body of the stomach, which is the major part of the stomach, and they intensify as they reach the antrum, which is the portion that tapers into the small intestine. Here's a picture to demonstrate what I'm talking about. Okay. The body of our stomach is right up in here. Here's the pylorus. Okay. What we're doing is forcing the stuff towards the end of our stomach. But the first couple hours, we're not going to really dump much out. I mean, this figure here, we're showing that it's not going to dump anything out because that pyloric sphincter is closed. So moving the stuff towards the pyloric sphincter is called propulsion. But just like if you throw something up against the wall, it's going to bounce back. That's the retropulsion. So what we're going to accomplish is a back and forth, kind of like a teeter-totter, mixing motion. And that's going to help us to form what's called chyme. Chyme is a mixture of our gastric contents, so our food and liquid, and then gastric juice. Okay. Now, this is great because it's going to allow for great digestion by the enzymes that like the really acidic environments. But as we know, salivary amylase will become inactivated. But it will stay active for a while, as I mentioned the other day. Okay. Now, where does this hydrochloric acid come from? Well, that's where these two figures on the next page come in. Okay. We secrete hydrochloric acid by way of parietal cells. Hydrochloric acid is abbreviated as HCl. We don't directly secrete HCl from the parietal cells. What we secrete are hydrogen ion and chloride, And then when they're in the lumen, they combine to form the hydrochloric acid. The hydrogen ion is produced in a reaction that we've seen before. Back when we talked about red blood cells, we learned about carbonic anhydrase. We also talked about this the other day with breathing. Remember the enzyme carbonic anhydrase joins water and CO2, forming an acid called carbonic acid. Carbonic acid that then dissociates to form hydrogen ion and bicarbonate. Here's the hydrogen ion, and it gets pulled into the lumen of our stomach through a active transport mechanism. And we know active transport requires ATP. As we pump hydrogen ion into the lumen of the stomach, we pull potassium in. We're not too worried about that. It's the hydrogen ion that we're focused on. The bicarbonate that was formed back in our reaction moves out into our interstitial space, which allows chloride to come in. Kind of how we talked about the chloride shift the other day. That chloride then gets pulled into the lumen of our stomach, where it then can combine with hydrogen ion and chloride. Now, this secretion of hydrogen ion and chloride occurs due to the presence of three things. The release of acetylcholine that binds to acetylcholine receptors on the parietal cells. Do you guys remember which division of the nervous system is involved with the release of acetylcholine? It's that division that produces that rest and digest response, our parasympathetic nervous system. Which should make sense. When we want the rest and digest activated, we want to secrete hydrochloric acid because that helps us rest and digest. The other two things that can stimulate the secretion of hydrochloric acid would be gastrin, that's released actually by the stomach itself, as well as histamine. Once again, those were produced by the ECL cells and the G cells of the stomach. Now, the hydrochloric acid activates lipase, gastric lipase, and pepsin. It inactivates salivary amylase. It does a couple other things. We've talked about how it will kill bacteria and other invading microbes. It also denatures the proteins in our diet. And as it states, proteins in food are denatured at low pH, which makes them more digestible. So I'm going to go ahead and list under proteins hydrochloric acid. It's not an enzyme, so I'm going to put that in parentheses. It's not an enzyme, but it helps denature the proteins. So it kind of helps with more of a mechanical digestion or physical digestion. Last thing I'm going to talk about is the way that our stomach functions and how it empties, which, by the way, takes several hours. So when somebody says they ate something and then a couple hours later or even a couple of minutes later they run to the bathroom and said, man, that ran right through me, they don't know what the heck they're talking about. They're full of, you know what. It takes a lot of time for food to run through you, as we're going to see. 2 to 6 hours, approximately, for our stomach to empty. Okay. As we said, while in our stomach, the food is churned through propulsion and retropulsion to mix the gastric juices with our food to form chyme. We eject chyme every 20 seconds into the duodenum in very small amounts, only about 3 milliliters at a time. The duodenum, by the way, is the beginning of our small intestine. Okay, as you can see here on the next page. Okay. We empty our stomach in response to both hormones and nervous mechanisms. But before we do that, and even before we fill our stomach, we start to secrete and start to get the stomach ready to start contracting. I like to think of our first phase, the cephalic phase, as like stretching before exercising. Getting your muscles ready to run. Well, we're not getting our stomach ready to run. We're getting it ready to receive food. We call it the cephalic phase because it involves your brain. The word cephalic refers to your head. Guess what's inside your head? Your brain. Okay. This is controlled by the vagus nerves. Once again, we know the vagus nerves are the major output of the parasympathetic nervous system. So this is a parasympathetic response that stimulates the secretion of gastric juice before food even reaches the stomach. So it can be triggered by the smell of food, the sight of food, just thinking about food. So right now you guys are all thinking about food. This is probably starting to occur. Okay. The initial taste of food, all can stimulate our stomach to get ready for arrival of food. That way, when the food hits the stomach, we can go right in and start digesting it. Okay. Now, the second phase is really where we amplify things up. Okay. We amp them up, control both neurally and hormonally. Neurally, it involves those receptors that we talked about the other day. Okay. Those enteric nervous system receptors sensitive to stretch and chemical concentration. Remember, we had stretch-sensitive receptors we talked about, and chemical chemoreceptor, or chemical-sensitive chemoreceptors. Okay. So as food enters our stomach, our stretch is going to occur in the stomach. Our stomach will stretch. It distends, as we call it. That activates the release of chemicals and begins the muscle contraction. So does a change in pH. pH changes as food enters our stomach, especially proteins. And that triggers the same thing. Release of gastric juice and waves of peristalsis. Okay. Hormones involve gastrin. Gastrin was that hormone that we release, or actually by the stomach itself, and it is released in response to distension of the stomach, partially digested proteins in the chyme, high pH of chyme due to food in the stomach, caffeine in the chyme, and acetylcholine released by the parasympathetic nervous system. Gastrin will stimulate the secretions of gastric juice, strengthen the contraction of the LES to prevent reflux, so it prevents the stomach from spilling, or the contents of the stomach from spilling back up into the esophagus, and then increases motility of the stomach, so it helps to mix things up faster and more, and then it's what stimulates the stomach to empty by relaxing the pyloric sphincter. Now, what does not occur is everything dumping out of the stomach all at once. As I mentioned, we only dump a little bit out at a time, and we can actually control how fast that occurs through the intestinal phase. The intestinal phase is a way to inhibit gastric emptying. Okay. Basically, what the small intestine can do is tell the stomach to slow its roll down if we're doing too much dumping or emptying. Or if we're emptying something that's really fatty. Okay. So let me read through and explain. Okay. Intestinal phase refers to inhibition of the stomach occurring when chyme enters the small intestine. Arrival of chyme in the duodenum stimulates the sensory neurons found in the small intestine that evoke a neural reflex that inhibits gastric motility and secretion. The presence of fat in our chyme also stimulates the secretion of intestinal hormones, inhibiting stomach motility. These help keep fatty chyme in the stomach for longer periods of time, allowing for the intestine more time to process that fatty chyme. So if it's something that's really fatty, or if we're dumping enough of it in that the intestines are kind of getting overworked, we feed back and tell the stomach to slow down. Okay. Does that make sense? So if you eat something really fatty, or a whole bunch of food, it's going to probably be more of like the 6 hours in the stomach before the stomach is completely empty. Figure on the left, I already talked about it. The figure on the right is showing you the feedback loop that exists when the stomach is stretched or when the pH changes inside the stomach, activating the response of gastric release, gastric juice release, including hydrochloric acid, muscle contractions, producing peristalsis, and then the response, as you can see, will be digestion, all that wonderful stuff. And then when the stomach is emptied, the feedback loop will be shut off. Now, instead of following our tract, we're going to take a trip to the side for a couple slides. We're going to talk about 3 organs that are not part of the tract, but are associated with the tract because of stuff that they produce. It includes our pancreas, our liver, and our gallbladder. We've talked about the pancreas before. Back when we talked about the endocrine system, we learned that the pancreas produced hormones like glucagon and insulin by way of cells called pancreatic islets. Those only represented 1% of the cells of the pancreas, which leaves us a whole bunch of other cells we call center cells that secrete what's called pancreatic juice. Pancreatic juice is juice from the pancreas. It's clear, it's colorless, made mostly of water, salts, sodium bicarbonate, and several enzymes. I'll talk a little bit more about the sodium bicarbonate in a second. Let's first talk about the enzymes. We've got pancreatic amylase. So we can add that now to our list of digestion for carbs. Trypsin is another protein digester. And then we've got pancreatic lipase, which actually happens to be our principal triglyceride digesting enzyme. I'm going to go ahead and add, because it's listed here, phospholipase. Because phospholipase, based on its name, it should not be a mystery or a surprise, breaks down phospholipids. We know, though, that phospholipids are not the primary form of fat in our diet, though, are they? What are the primary form of fat in our diet? Not phospholipids, but triglycerides. Okay. But since phospholipids are a form of fat, I'm going to go ahead and keep that in my list of enzymes that digest fats. The other day I mentioned that there were 4 organic compounds that we talked about previously. Carbs, fats, and proteins. Those are all listed over there on the board. What was the 4th that's not listed? Nope. What is inorganic? It's where DNA and RNA fall in. Those nucleic acids. These 2 enzymes help digest DNA and RNA, which we will ingest. DNA, which was short for deoxyribonucleic acid, is broken down by deoxyribonuclease. RNA, short for ribonucleic acid, is broken down by ribonuclease. Am I going to worry about you knowing those? No. Okay, because I don't have a list of nucleic acids over there. They're not a very important component of our diet. Okay. Now, stimulation of pancreatic juice release is controlled by hormones once chyme, or food from our stomach, begins emptying into the small intestine. A trigger is a drop of pH of the duodenum below 4.5, which causes the release of secretin by glands of the small intestine. Okay. Secretin is a hormone that stimulates the pancreas to release bicarbonate. Sodium bicarbonate is very important because it will buffer that hydrochloric acid coming into our small intestine. I said that the stomach was really well built for hydrochloric acid. Our small intestine is not. It doesn't have the mucus lining like the stomach does. But it does have sodium bicarbonate in it because of the pancreas. And so the sodium bicarbonate will neutralize the acidic kind. Another trigger is the fat and protein content of chyme, resulting in the release of cholecystokinin, CCK, from our small intestine. CCK stimulates the pancreas to produce and secrete those enzymes that we just talked about. Both secretin and CCK also stimulate the liver to produce bile, which is a very important fat-digesting chemical. I'll talk more about it in a second. Should be no surprise, major nerves innervating the pancreas are the vagus nerves, which provide parasympathetic outflow. When our rest and digest response is active, the pancreas is active. When the sympathetic nervous system is active, the pancreas is inactive. Our next accessory organ of the digestive system is a very important organ, and it's the largest organ of our body, actually. Inside of us, that is. Your skin is the largest totally, but it's on the outside. Your liver, a bilobed organ made up of smaller lobules, does a whole bunch of different things. One of the most important things it does is produces bile. As I just mentioned, bile is a very important chemical involved with fat digestion. Now, bile should not be mistaken as an enzyme. What bile does is emulsifies fats. And I've got a figure that will demonstrate what emulsification is. It's here on the page of the gallbladder. I'll go ahead and let you guys write out the page here that I'm on, and then I'll explain what I'm talking about. Our gallbladder and our liver are very closely associated with one another. Your gallbladder actually sits on the bottom of and kind of behind your gallbladder. If you look at the figure here on the next page, it can show where the gallbladder is located in relationship to the liver. Okay. Your gallbladder is basically just an extension of your liver, and it helps to store and concentrate bile that the liver actually is producing. Down at the bottom, I can show you what happens when we release bile and dump it into our small intestine. If you think back to the beginning of the semester, we learned that fats don't get along very well with water. Well, a lot of the contents of your stomach and intestines will be water, okay? Whether you're drinking it directly or if it's part of your food. Okay? So when the fat is in your stomach and the small intestines, the fat's going to want to clump together with other fat and form large globules of fat. Those large globules of fat are really hard to digest. Okay. Think of it like a big chunk of ice. That melts really slowly, but if you break it apart into smaller pieces, the smaller pieces melt much more rapidly. That's basically what bile salts, which are a component of bile, do. They take a large lipid globule and chop it up into smaller pieces. So bile helps to perform mechanical or physical digestion. Okay, it's not a chemical breakdown There is no bond breakage here. It's a big piece to smaller pieces. Okay. Now, on top of the bile salts, there's other parts of bile. Okay. It includes water, cholesterol, lecithin, biopigments, several ions. Some of the stuff is actually just going to get lost in our feces. Okay, so it's an excretory product. Okay, some of it is actually important for that emulsification process. Okay. Each day, our liver produces about 1,000 mils of it. It's about a liter, so that's a very significant amount. Okay, it's a yellow, brownish, or olive green liquid. Has a pH of 7.6 to 8.6. Okay. Bilirubin is a very important biopigment formed from the recycled parts of the hemoglobin. We actually talked about that before. On top of producing bile, your liver does a whole bunch of other things. Okay. For those of you who are taking anatomy or have taken anatomy, which is every single one of you in here, we talked about in anatomy the hepatic portal vein. So for those of you who are in anatomy, this is something we just talked about last week. The hepatic portal vein was a vein that directly connected your liver with your intestines, your stomach, your spleen, and some other structures. Okay. Well, as we're going to see, most of the stuff that we absorb is absorbed in your small intestine. And if we're talking carbs, proteins, and fats, like we are, those things can get directly carried to your liver, which if you see here, our liver is responsible for processing and metabolizing those three fuel nutrients. Okay. We're going to talk in greater detail about some of these things, especially carb metabolism. Okay. Our liver can help maintain normal blood glucose or blood sugar levels. When our blood glucose levels are low, what we call hypoglycemia, we can break down glycogen to glucose that's stored in our liver to release it into the bloodstream. We call that process glycogenolysis. It can also convert amino acids, lactic acid, and other sugars into glucose, a process that we call gluconeogenesis. When our blood sugar levels are high, what we call hyperglycemia, we can perform glycogenesis, conversion of glucose into glycogen. These are words that we've heard before. These are words we're going to look at more in our next lecture starting on Friday. Okay. Our liver also metabolizes our lipids. Some of our hepatocytes store triglycerides. Others break down fatty acids to generate ATP. We synthesize lipoproteins in our liver. Lipoproteins are a lipid fat protein hybrid. Synthesize cholesterol and use cholesterol to make biosalts. We deaminate amino acids in our liver. Deamination is where we remove an amino group. This is something I will talk more about on our next lecture, Okay. That's important because then we can use those amino acids to make ATP. Okay. While this occurs, we actually will end up forming a toxic chemical called ammonia, but our liver will convert that ammonia to urea, which can then be lost through urine. We talked about plasma proteins, fibrinogen, albumins, globulins. Most of those are made in our liver. A lot of people are very aware that habitual drug users and alcoholics will destroy their liver because your liver is what processes those drugs and hormones. Okay. Causes cirrhosis, especially alcoholism. Okay. The hormones include the thyroid hormones and steroid hormones are processed by our liver. Excretion of bilirubin, we talked about that a little bit ago. Okay. Secreted into bile from our liver, comes from breakdown of hemoglobin, synthesis of bile salts for emulsification, Storage of things like vitamins and minerals, phagocytosis of aged red blood cells, white blood cells, and even platelets. Remember, that's like Pac-Man running around chewing stuff up. And then activating vitamin D that we get from the sun, along with our kidneys and our skin. Any questions on any of those organs? Now, the figures here on the last page, and not the last page of the lecture, but the last page about those 3 accessory organs, demonstrates the intertwining that exists between those 3 organs. Okay, so here in our liver, we can see we have 2 major lobes, right in the left lobe. Those provide bile into these 2 bile ducts. Those 2 bile ducts, or hepatic ducts as they're called, form what's called the common hepatic duct. And along with the cystic duct, those unite to form a common bile duct. That's going to channel bile down, and it actually joins with the pancreatic duct, which is the major duct carrying pancreatic juice. And therefore, pancreatic juice and bile both are dumped into the same spot in our duodenum. And so we don't do any direct digesting of anything between these 3 organs, but we help out our small intestine in the digestion of those various things. So let's now talk about the small intestine, which I always say is where most of the magic happens. And by that I mean most digestion and absorption of nutrients. Now, our small intestine gets its name not for its length, because it's actually the longest part of our tract. It gets its name because it's not very wide in diameter. It is a long, winding, twisting tube which provides for a large surface area for digestion and absorption. Okay. But on top of that, we have 3 special features that increase the surface area by a factor of 600. So 600 times more surface area provided by these 3 structures. Okay. Together, the total surface area of the small intestine is approximately 200 square meters, about the size of a tennis court. It's an incredible amount. So here on the next page, we can show what those 3 structures are. Okay. Over here on the right side of our page, we see a figure. We're zooming in at a small section of the small intestine. And if we open it up, we see that it's not smooth on the inside. Kind of how like the stomach had depressions. Instead of depressions, these are extensions. So these point outward into the lumen. Okay. These are little finger-like folds. You can see if we zoom in at one of them what they look like. Okay. Called circular folds or plicae, about 10 millimeters in length, okay, which is a centimeter. Okay. So you can actually see these with the naked eye. Okay. About 1/2 to 1 millimeter in length are little villi, which if you look closely enough at this image down here, we see little ruffles. Those little ruffles are what this is. Okay. These are the little finger-like extensions called villi. Okay. Villi is the plural form for villus, which is one of these that's shown here. Okay. On Friday, we'll talk about the inside of a villus, where you find blood vessels and lacteals. Today I want to talk about the cells that make up a villus, which includes cells that have microvilli, which, if you don't remember, at the beginning of the semester we talked about microvilli, are the extensions of cells that look like little fingers forming a brush border. And you can see they're labeled up there at the top, right on our absorptive cells. This allows for great absorption, where stuff is brought into the cells, ingrate secretion, where stuff is released from cells. Your absorptive cells do both of those things. Obviously, they absorb, hence the name, but they also produce some enzymes. Okay. And we'll talk about that here in just a second. Those enzymes are called brush border enzymes. Okay. So we've got folds on folds on folds, basically, on the inside of our small intestine. And mainly we're focusing here on just the mucosa, the inner lining of our small intestine. Okay. We've also got goblet cells that secrete mucus to help protect and lubricate the small intestine. They don't really play a huge role. Not quite as important as the absorptive cells. And then we've got intestinal glands, which include a whole host of cells that secrete the intestinal juice. About 1 liter to 2 liters a day. A clear yellow liquid, which contains water and mucus and is slightly alkaline, pH of 7.6. Together, it with the pancreatic juice helps provide a liquid medium for absorbing substances from chyme. Several enteroendocrine cells are found in the crevices of our intestinal glands and include Paneth cells, which are the ones that secrete that lysozyme that we've talked about this semester, antibacterial enzyme. CCK cells that secrete CCK, cholecystokinin, and S cells that secrete secretin. Well, we just talked about cholecystokinin and secretin. Those two hormones help to activate not only the pancreas, but also the liver. Okay. The submucosa is where you find duodenal or Brunner's glands that secrete an alkaline mucus that, along with that sodium bicarbonate from the pancreas, neutralizes the gastric acid in the chyme. So that was related more to chemical digestion. Your small intestine also helps perform mechanical digestion through two processes. Segmentation, which I compared the other day to like grabbing a toothpaste tube in the middle of the tube, like my lovely wife does. Okay. Segmentation will mix the chyme with digestive juices and bring them, or the particles of them, into contact with mucosa for absorption. Okay. Remember, segmentation is not a forward movement, it's a backwards and forward movement, so it will not push the contents of the tract or along the tract because that occurs through the other process. This is a special form of peristalsis called the migrating motility complex. Okay. This special form of peristalsis begins after most of a meal has been absorbed. Begins in the lower portion of the stomach, pushes chyme forward along a short stretch of small intestine before dying out. The migrating motility complex, or NMC, slowly migrates down the small intestine, reaching the ileum in 90 to 120 minutes. Okay, so an hour and a half to 2 hours. Then it begins all over again, and so chyme, as a result, will remain in our small intestine for up to 5 hours. Okay. And if your food in your stomach was in there for 6 hours, now we're up to 11 hours. Now that's max, probably. It could be as low as 2 in the stomach and as low as 3 in the small. So it could be as few as 5 hours. But we're still not even pushing that chyme quite yet out of us because we still got the large intestine. Now, before we get into the large intestine, we have to talk about digestion and absorption in greater detail. And what I did here for the next 3 slides is broke down our 3 fuel nutrients and talked about them individually. Kind of like how I have them in separate columns over there on this side of the room. We've already talked about the 2 primary amylases that help with digesting carbohydrates. Salivary amylase, which becomes activated in our mouth but deactivated in our stomach. And then pancreatic amylase, which is working in our small intestine. Okay. Both of those amylases help take our carbohydrates which are principally going to be in polysaccharide form, and break them down into disaccharides, which is where you have two monos bonded together. We can absorb disaccharides, but we prefer to absorb monos. And that's where three brush border enzymes come into play. We have sucrase. Guess what it breaks down? Sucrose. Sucrose, which we learned earlier in the semester, is a fancy way to describe table sugar. We've got lactase. Guess what it breaks down? Lactose, which is found in dairy, milk, cheese. So people that have lactose intolerance don't produce enough of this, and therefore have trouble digesting dairy. And then maltase, which digests maltose. Malted sugar. Now, we don't digest everything. Carbohydrate, that is. Cellulose is actually an indigestible polysaccharide. Okay. We can't digest it, therefore we can't absorb it. Okay. Cellulose is the plant wall, or plant cell wall, in the cells of, obviously, plants. Okay. So a lot of vegetables are very hard to digest. Corn is a very good example of this. You eat corn. Here in Kansas, we eat a lot of that. It comes right out looking exactly like it did before. Okay, it's a great example. That's actually really good for us. Fiber is what we a lot of times will call indigestible carbs. Actually speeds up the movement of stuff through our tract, which can be dangerous if you're not used to it. Not dangerous as it's going to kill you. Dangerous like you can poop your pants. Okay. Which might you'd rather die than do that. But it helps keep things regular and actually will lower your risk for like colon cancer. Okay. But the ultimate goal for the things that we do digest, break them down into monos. That way they can then be absorbed. Proteins. Remember, proteins are made up of amino acids put together in chains. We must break them down into either dis, tris, or singular amino acids where they then can be absorbed. Okay, dipeptides are 2 amino acids, tris are 3s, and then a singular would just be 1 like you see here. Begins with pepsin produced in our stomach, aided by hydrochloric acid, even though it's not listed. Also in our stomach, because that helps denature the proteins. Continues in our small intestine with trypsin from your pancreas. And then you've also got, and I didn't list it, within our pancreatic juice you have chymotrypsin, carboxypeptidases, and elastase. It is completed by 2 what we call peptidases, which are brush border enzymes. We've got aminopeptidase and dipeptidase. I'm not going to worry about any of the other pancreatic enzymes. The only one I want you to know is trypsin. But I do want you to know the 2 peptidases. We will talk about how we absorb the amino acids on Friday. Your fats involve the lipases, as we know. Lingual lipase, produced in our mouth, not active until it reaches our stomach. Gastric lipase, active in our stomach, produced by our stomach. Principal fat digesting enzyme is pancreatic lipase. I also added phospholipase because it digests phospholipids, and then bile because it helps emulsify. Even though phospholipids are not the major component of our fat in our diet, they are important. What are the most abundant lipids in our diet are triglycerides. Here we can see what a triglyceride looks like. If you can't remember, they are a 3-carbon glycerol. Attached to them are 3 fatty acid tails. In order to absorb them, what we need to do is break down those into the 3-carbon glycerol and then release the fatty acids. Okay. And so the process of digestion and then absorption is very complicated with fats. It involves structures called micelles, which are formed when bile salts surround the free fatty acids and monoglycerides. And then we'll also talk about some other stuff related to them first thing on Friday. Table 24.5 is a nice summary of the enzymes involved with digestion. So if you want to study it, it might be helpful. Of course, I wrote over there on the board the main things that you needed to know. We'll pick up first thing on Friday with absorption. This is where the stuff enters the body into our bloodstream or lymphatic fluid.